안녕하세요. 저는 하버드 의과대학에서 MD, PhD 과정을 수료 중인 이석준입니다. 오늘 저는 버나드 사바티니 교수님 연구실에서 PhD 과정 동안 연구했던 주제에 대해 발표하려고 합니다. 발표 제목은 Dopamine Dependent Modulation of Striatal PK Activity During Reinforcement Learning입니다. 제가 과학 주제 발표에 있어서 한국어가 서툰 관계로 주 발표는 영어로 진행하겠습니다. So let's start with a simple diagram. One of the fundamental tasks a brain has to solve is to forming an association. When an animal performs an action, it usually receives some kind of feedback from the environment, and if the outcome is positive, the animals may want to repeat that action in the future. To do so, the animal has to form association between the action and the outcome from the environment. In the learning literature, we call this type of learning reinforcement learning because a positive outcome is reinforcing the action that presumably resulted in the outcome. One dominant theory in neuroscience regarding this reinforcement learning is a real prediction error theory, where there's a signal that encodes a difference between the actual outcome and the expected outcome that can act as a teaching signal. And this teaching signal is a very effective way of learning because it will only emerge when there is a discrepancy between the outcome and the expectation, suggesting a need for learning something new. Interestingly, there is a classic study by Schultz who identified a group of midbrain dopaminergic neurons whose activity looks like this real prediction error. For instance, for a naive animal, there is an increase in dopaminergic neuron activity at the time point of reward, which is consistent with the positive real prediction error, where there was a positive actual outcome with zero expectation. On the other hand, for the trained animal, the dopaminergic neuron activity now shifts to the cue, and for the actual time point of the reward, there is a minimal activity of the dopaminergic neuron, which is consistent with kind of a net zero prediction error where the actual outcome was matched with the equal amount of expectation. Furthermore, when we actually omit the reward for the trained animal, what we see is a dip in dopaminergic neuron activity below the baseline, which is consistent with the negative real prediction error where there was no actual outcome with a positive outcome, therefore negative net error. Then what does dopamine do? One major projection target of the midbrain dopaminergic neuron is striatum, which is the input region of the larger structure called basal ganglia. Striatum contains two major subtypes of neurons, direct pathway neurons that expresses D1 receptor and indirect pathway neurons that expresses D2 dopamine receptor. With this knowledge, many people came up with this model that dopamine acts as a teaching signal to direct and indirect pathway neurons, inducing some kind of plasticity in these cells, allowing the dopamine and basal ganglia dependent learning to happen. Then what is the mechanism of dopamine action on striatal neurons? In other words, is there any biological substrate that represents learning in these neurons? Before we delve into this question further, I want to clarify a few things for today's talk. I will focus on VTA dopaminergic neuron instead of SNC dopaminergic neurons. Also, I focus on the nucleus accumbens, which could be anatomically distinguished from dorsal striatum. And because the projection patterns of the nucleus accumbens and the dorsal striatum are distinct, I will avoid using a term direct and indirect pathway neurons. Rather, I will use a term D1 receptor expressing SPNs or D2 receptor expressing SPNs to describe two types of neurons in the nucleus accumbens expressing two different types of receptors. The hint for the possible downstream mechanism of dopamine action on striatum can be found in dopamine-dependent intracellular pathways. D1 receptor is a GS-coupled receptor. Therefore, dopamine binding to D1 receptor results in increase in adenylyl cyclase activity, level of CMP, and increase in PK activity. In contrast, D2 receptor is a GI-coupled receptor. Therefore, dopamine binding to D2 receptor suppresses the activity of adenylyl cyclase, level of CMP, and suppresses the PK activity. pKa is a particularly interesting downstream target of dopamine because it has been studied and implicated in the plasticity by many studies via changing conductance of various ion channels and also changing a transcriptional profile. With this knowledge of intracellular pathways, we can now come up with this hypothesis for dopamine action on D1 and D2 receptor SPNs during learning. When there is an unexpected reward, this causes the increase in VTA dopaminergic neuron activity, increase in the level of dopamine in the nucleus accumbens, and that may actually bidirectionally change the activity of the pKa in D1 and D2 receptor neurons. And this upregulation of D1 receptor SPN's PK activity and downregulation of D2 receptor neurons PK activity may be a substrate for reward-based learning. However, it has been difficult to directly test this critical link between dopamine and SPN PK activity because it was difficult to measure the activity of intracellular pathway in vivo. And today I will talk about how we tackle this challenge. So this is the outline for today's talk. I will first talk about the technique to measure dopamine in vivo, and then we will talk about the PK activity in vivo. Then I will show you the dopaminergic neuron activity and dopamine release pattern during learning. And then I will talk about the PK activity during learning, then trying to show you the causal relationship between the two. And then I will end with the causal relationship between the PK activity as well as the learning. During my PhD training, I had an amazing collaboration with the Lin Tian's group in UCSD who developed an optical sensor for dopamine. In brief, the D-light, the dopamine sensor, is an optical sensor based on fusing the dopamine receptor to a green fluorescent protein. And this sensor changes its fluorescence intensity when dopamine binds to the receptor domain of the sensor. Because this sensor is a protein-based sensor, we can express the sensor using a virus that encodes a protein by injecting the virus into the rodent brain stereotactically. Then we can implant the optical fiber into the brain to relay the signal through the flexible fiber, which allows the animal to behave without head restriction. And this was a major advance in the field of dopamine-related neuroscience field because it made us much easier to measure the dopamine and also combine this optical method with other optical method to measure different part of the neural system. Now I will talk about how we can measure the PK activity in vivo. For in vivo measurement of PK activity, we have developed a new method adapting a fluorescence lifetime technique that is often used for two-photon microscope. In particular, we are using a sensor called FleaMaker developed by Yao Chen, who was a postdoc in our lab. And this sensor works as following. Without phosphorylation of the pKa substrate domain of the sensor, the donor and the receptor molecule are far apart. And the donor receives the excitation energy and emits photons in fluorescence. When the endogenous pKa phosphorylates the substrate domain of the sensor, now this conformational change brings the donor and the acceptor together. And at this point, the donor can lose its energy to the acceptor instead of emitting the fluorescence light. The donor and acceptor molecules could get closer, and the donor molecule will now lose its energy to the acceptor molecule. And because of this energy transfer, the fluorescence of the donor will decay much faster in this conformation. So now when we look at the average fluorescence lifetime, or which shows us how fast the fluorescence decays, we can see that when there is a low PK activity, the fluorescence will decay much slower. And when there's high PK activity, the fluorescence of the sensor will decay much faster. And by measuring this decay rate of the fluorescence, we can measure the endogenous activity of the pKa. So how do we actually use this sensor in vivo? We took advantage of FleaMaker and a modified fiber photometry system that can detect fluorescence lifetime of the sensor from the brain of awake, freely moving animals. Briefly, we expressed the FleaMaker using a virus, as I just mentioned previously, with a D-light sensor. And after expressing this sensor, we can also relay this optical signal from the nucleus accumbens using the fiber optics. And this signal will be now cast by a hybrid PMT, which is basically a photons or the light sensor, and then processed by an electronic called time correlating single photon counting machine, which can provide us with a lifetime histogram showing how fast the fluorescence decays. To test the fluorescence lifetime photometry technique, we first measure the D1 receptor agonist response of the FleaMaker, the PK sensor expressed in D1 receptor SPNs. In all FleaMaker graphs that I'm going to show you today, a negative change in the fluorescence lifetime is depicted as the uprising slope because this represents the increase in PK activity. Here, the IP injection or intraperitoneal injection of D1 receptor agonist to awake behaving animal resulted in a marked increase in PK activity in D1 receptor SPNs, which is consistent with our understanding of D1 receptor being GS coupled receptor. I also want to draw attention to not only the direction of the PK activity change, but also the kinetics of the response that lasts thousands of seconds. And this is likely representing the slow pharmacokinetics of the IP-injected drug that takes a relatively long time to reach the target brain region and to degrade, given that the later experiments have shown that ability of the sensor to show a faster change in PK activity. On the other hand, the mutant sensor in red, where we mutate the phosphorylation site of the sensor, exhibited a minimal change in fluorescence lifetime, suggesting that this change is likely driven by the phosphorylation of the PK substrate domain of the sensor. We further investigated the effect of other dopamine receptor drugs, which showed a change in PK activity consistent with the prediction from D1 receptor and D2 receptor modulation of PK activity. I want to point out a few things here for the D1 receptor antagonist and D2 receptor antagonist, which resulted in slight inactivation or the activation of PK activity in D1 receptor or D2 receptor SPNs, respectively. And this change in PK activity is possibly due to the effect of basal tone of dopamine in the downstream pathway, although we cannot exclude the possibility of inverse agonist activity of these antagonists. We further tested our technique to see if we can actually detect a possibly faster change in PK activity induced by natural stimuli. To do so, we measured the PK activity in response to a food reward. And consistent with the prediction from the action of D1 receptor on PK activity, D1 receptor neuron PK activity has a significant increase at the time point of reward consumption. And I want to point out that now the kinetics of the SPN PK activity is now in the order of 50 seconds, which is very different from what we have seen in the pharmacokinetics experiments of the D1 receptor agonist that has lasted thousands of seconds. So far, I have shown you that fluorescence lifetime photometry, or FLIP, can detect bidirectional change of PK activity in D1 receptor or D2 receptor SPNs. Also, FLIP can reveal the in vivo pharmacokinetics of GPCR drugs. Lastly, the dopamine receptor antagonist experiments provide in vivo evidence for possible basal tone of dopamine acting on dopamine receptors, although we cannot rule out the possibility of inverse agonist activity of antagonists we used. With knowledge that PK activity in SPN change induced by a food reward is in the order of tens of seconds, we developed a behavior paradigm that works on a slow time scale, which will help us to assign signal patterns to distinct behavior time points. The paradigm is a visual cue guided reinforcement paradigm that is based on giving a food reward to hungry animals. During the session, the mice are allowed to freely move inside the behavior box during the inter-trial interval of 120 seconds. After that, the mouse can enter this artificial green zone and stay there for 3 seconds to initiate the trial, which is signaled by the onset of the LED. After LED onset, now the animal has 5 seconds to enter this blue zone, which is closer to the LED as well as the receptacle that dispenses the reward. Once it enters this blue zone, now it has to stay for another 3 seconds until it gets the dispensing of the pellets. At any point, if the animal fails to perform the specified action, the LED will turn off, signaling the ending of the trial. In terms of training the animal, I have trained the animal for 11 days, and after 11 days, they experience 2 additional sessions with specific manipulation. For day 12, rewards are omitted from 25% of successful trials randomly, and for day 13, LED is turned off for the full session, although the animals can still perform a sequence of action purely based on timing. So this is an example of a success trial, and I will show you the video. The mouse first enters this trigger zone that's opposite from the receptacle, stays there for 3 seconds, the LED turns on, the mouse enters this receptacle zone, stay there for 3 seconds, and gets the reward. This is an example of one type of failure trial, which I will call entering failure. The mouse first enters a trigger zone, stays there for 3 seconds, LED will turn on. The animal does not enter this receptacle zone within 5 seconds. That terminates the trial. This is an example of another type of failure trial, which I will call occupancy failure. The mouse first enters the trigger zone, stays there for 3 seconds, LED turns on, it goes to the receptacle zone on time, but prematurely exits, and therefore the LED will turn off without the reward dispensing. So can animals learn this task? Here I am showing the evidence of learning. The animals gradually increase the success rate close to 90% across 11 days on average. And this increase in success rate comes from the large reduction in receptacle zone entering failure rate, as well as a small reduction in occupancy failure rate. Furthermore, animals begin to stay in the trigger zone longer on average while reducing their time in sitting near the receptacle. With this knowledge that animals can learn this slow timescale task, we decided to trace a dopamine signal and its effect on downstream neuron during its behavior. First, we express JRCAM1B, which is a red calcium sensor and BTA dopaminergic neuron to monitor both soma and terminal activity of dopamine neurons. Second, we express D-light, which is an optical dopamine sensor to monitor dopamine release in the nucleus accumbens. Lastly, we express FLIMACAR, the PK sensor, to monitor the PK activity in either D1 or D2 receptor SPNs. So now I will talk about the patterns of dopaminergic neuron activity and dopamine release pattern during learning. To simultaneously monitor dopamine neuron activity and dopamine release, we took advantage of multi-channel photometry system, which allowed us to measure both D-light and JRCAM1B signal in the nucleus accumbens, as well as a JRCAM signal in the VTA. Therefore, we can simultaneously monitor both the release of dopamine, the terminal activity of the VTA dopaminergic neuron, and the soma activity of the VTA dopaminergic neuron. So let's start with dopamine. In this heat map, the warmer color stands for higher D-light signal. Each row is an individual trial from multiple mice, which can be identified by color code on the mouse ID column on the right. Lastly, red line or the dots stand for the different time points within the task, including trigger zone entry, LED onset, receptacle zone entry, dispensing of the pellet, and then receptacle entry for the reward consumption. Trials from all mice are sorted by the receptacle zone entry point. First, let's focus on the dopamine release aligned to the reward predictive LEDQ, or DL. For the beginner mice, we can see there is a minimal response of D-light to LEDQ. But when we look at the expert animal, we see a high signal that's being aligned to the LED onset. And that pattern is more obvious when we look at the average graph, where the green graph represents the expert animal, the red graph represents the beginner animal. In contrast, we can see kind of the opposite trend for reward signal. The reward signal that's being aligned to either D or R are much higher on the beginner animals and much weaker on the expert animal. And this trend is a little bit more obvious when we average out the graph here. And the reason the reward response starts before the actual consumption of the reward or entry to the receptacle is most likely because animal now shows a response to the click sound of the dispenser itself, and that's supported by the fact that the increase in D-light signal close to this R actually starts right after the dispensing of the pellet, which causes a click sound. We can look at the trend of dopamine signal in another way by plotting the average response of the mice to cue or reward across days. And as you can see, the cue response gradually increases and the reward response gradually decreases across days. Furthermore, this pattern is preserved across all three signals, including soma, VTA dopaminergic neuron activity, measured by VTA JRCAM signal, and the terminal dopaminergic neuron activity measured by nucleus accumbens JRCAM signal. Then what about the reward omission? In this heat map, D stands for dispensing of the pellet, or the expected time point of the pellet dispensing. And when we omit the reward for mice that have completed the trial successfully, we now see a dip in dopamine below the baseline starting at the time point of pellet dispensing, or expected time point of pellet dispensing, consistent with the idea of negative RPE. Furthermore, we added an additional trial type, fully trained animal, where we omitted the LED completely while allowing the mice to complete the task purely by self-driven action sequences. And indeed, the success rate of all mice plummeted and the LED was omitted, suggesting that expert mice do use the LDQ to guide their behavior. However, there are a few trials they succeed to receive reward, and I'm showing you only 60 trials here. And in this case, the response to the reward was much larger than the regular success trial, presumably because they had a lower expectation for rewards, which is consistent with the idea of positive reward prediction error. In addition to confirming that dopamine signals are consistent with predictions from the reward prediction error, we can also test additional hypotheses of how correlated these signals are in real time because we are measuring all three signals simultaneously. On the left, I'm plotting the three different signals from a single session from one example mouse. And green line stands for the behavior time points for success trials. Orange lines stand for behavior time points for reward omission trials. And by looking at the general patterns grossly, we can see these signals are very much alike. And given that the kinetics of these three signals are quite different, as you can see on the magnified graph here, much slower kinetics on the top and much faster kinetics on the bottom, we can quantify the correlation across signals by first deconvolving the signals into population spikes and looking at the correlation between these deconvolved signals. And when we did that comparison, we saw still a moderate correlation between the cell body activity and dopamine release at the level of the moment-to-moment correlation. In summary, I have shown you that there are multiple pieces of evidence that support that dopamine neuron and dopamine release pattern are consistent with the reward prediction error across learning as well as within the trained states. For example, we saw reduced reward response across learning, increased cue response across learning, dip following the reward omission in trained mice, and larger response for unpredicted rewards in trained animal. Also, I've shown you that dopamine neurosoma activity and release of dopamine in the nucleus accumbens maintain a moderate correlation during behavior. Next, I will talk about the patterns of PK activity during learning. To simultaneously monitor dopamine release and the PK activity in SPNs, we had to express D light on one hemisphere and FLEMA, the PK sensor, on the other hemisphere. And the reason for this schematic is because these sensors are unfortunately both green sensors. But to test the validity of these two hemisphere experiments, we have confirmed that dopamine release in this non-lateralized learning task is very symmetric in a separate experiment of expressing D light on both hemisphere and looking at their correlation, which showed a pretty good correlation across all behavior. Okay, now let's take a look at how dopamine is related to PK activity. As we have seen before, there is a large increase in dopamine at the time point of the reward. On the other end, there is a minimal response to cue for both success and failure trials. How about the PK activity in D1 receptor SPNs? As we have expected from our simple palette experiments, there is a marked increase in PK activity at the time point of the reward consumption. On the other hand, SPN PK activity does not really change right at the time point of the LDQ. And this is a little bit more clear when we just look at the failure trials where there is only the cue without the reward. And this pattern strongly suggests that dopamine may be driving this strong activation of D1 receptor SPN's PK activity because PK activity in D1 receptor SPNs are only really changing at the time point of significant dopamine increase. Now let's look at how training changes the patterns of neural signals. Here, I'm depicting the beginner trials in red and expert trials in blue. First, the dopamine peak at the time point of the reward is much smaller for the expert animals compared to the beginner animals. On the other hand, the dopamine now has a marked increase at the time point of cue for the expert animals, consistent with the reward prediction error. Furthermore, there's a significant dip in dopamine at the time point of the expected reward for the failure trials, which were not present in the beginner animals in red. How about PK activity in SPNs? You can see that D1 receptor SPN's PK activity now has shifted from the receptacle entry time point to the LADQ. And because the PK activity change has a much slower kinetics than dopamine release, it is a little bit more difficult to parse out how much of this activation during the success trial for expert animals is driven by the LDQ or the reward itself. However, by comparing the success trial and the failure trials of the expert animals, so these two blue graphs, we can see there's both the LDQ component as well as the reward component in this activation of PK activity in D1 receptor SPNs. What about the D2 receptor SPNs PK activity? D2 receptor SPNs PK activity was not strongly modulated in general except in one case. And for expert animals, D2 receptor SPNs PK activity increased significantly at the time point of the predicted reward delivery for failure trials, which shows a possibility of this dopamine dip-driven activation of PK activity in D2 receptor SPNs. To formally test how expectation is changing both dopamine release and PK activity in SPNs, as we have seen in the failure trials in the previous slides, we omitted the rewards from successful trials for expert animals. Here, the red represents the expert animals' response in rewarded trials, while blue represents the expert animals' response in unrewarded or reward omission trials. Consistent with the results in the failure trials, the reward omission resulted in marked reduction in dopamine release. Also, for the D1 receptor SPNs, there was a smaller activation of PK activity, which is consistent with the lack of the second peak of dopamine induced by the reward. Due to slow kinetics of the lifetime change, it is still difficult to say whether this actual dip in dopamine has additional effect on D1 receptor SPNs PK activity just from this graph. On the other hand, D2 receptor SPNs, there was a marked increase in PK activity, presumably due to the dip in dopamine level. So far, I have shown you that there is an asynchronous activation of PK activity in D1 receptor and D2 receptor SPNs during behavior. D1 receptor SPNs PK activity increases significantly at the time point of the reward or reward predictive cue when there is a dopamine increase. For D2 receptor SPN PK activity, it increases significantly only for the trained animals when there is dopamine dip. So far, I have just shown you the correlation between dopamine and PK activity in SPNs. And now let's talk about whether they are causally related. To determine the SPN PK modulation that is correlated dopamine level is actually driven by dopamine receptor. We pharmacologically block the receptor with IP injection of D1 or D2 receptor selective antagonist before behavior starts for expert animals. Also, for this particular set of experiments, we are passively providing either LDQ or free pellets because dopamine receptor antagonists can perturb the performance of animals. And this pretreatment of D1 receptor antagonists resulted in a near complete blockade of LDQ-induced PK activity change, as well as a food reward response. The pretreatment of the D2 receptor antagonist had a similar effect. It completely blocked the reward omission response of the D2 receptor SPN PK activity following the LED due to the reward omission. And this suggests that basal binding of dopamine to D2 receptor is required for the dip in dopamine to cause a PK activation in D2 receptor SPNs. Furthermore, the pretreatment of D2 receptor antagonists did not really change the small reduction in D2 receptor SPN's PK activity following the reward, suggesting that this response is likely to be dopamine receptor independent. Then, to test the sufficiency of dopamine action on PK activity, we relied on optogenic manipulation of VTA dopaminergic neurons. To bidirectionally modulate VTA dopaminergic neuron, we either express a crimson R, which is an activating opsin, or GTACR2, which is an inactivating opsin in VTA dopaminergic neurons of the VTA dopaminergic neurons of that cree mice. You can see on the right, the activation of the dopamine neuron using crimson increases the level of dopamine in vivo, while inhibition of dopamine neuron via GTACR2 results in significant reduction of dopamine below the baseline. And with this tool, we now can test if the dopamine level change can affect the PK activity directly by expressing frelime ACAR, the PK sensor in the nucleus accumbens. And to selectively express the sensor in the D1 and D2 receptor SPNs, we now use a double transgenic animal that cree crossed to D1 or Adora cree. All right, let's now take a look at SPM-PK activity change in D1 receptor neurons. We try to match the amount of dopamine release by optogenic activation of dopamine neuron to reward-induced dopamine release. And with this protocol, we have found that dopamine neuron activation was sufficient to increase the D1 receptor SPM neurons PK activity. For the inactivation of dopamine neuron, we have not found any significant change in D1 receptor SPM PK activity, even with the longest dopamine neuron inactivation. And this suggests that there is a minimal binding of dopamine on D1 receptor on baseline. On the other hand, the opposite trend has been found in D2 receptor SPM's PK activity. When you activate dopamine neuron using crimson, there is almost no change in PK activity, and therefore a small reduction in PK activity in D2 receptor SPMs during behavior again is most likely due to a dopamine receptor independent. For dopamine neuron inactivation, we try to match the GTACR activation so that the dip in dopamine caused by optogenic manipulation is similar to that of the reward omission response. In black, I'm plotting the reward omission response after the reward predictive cue, which has caused the preceding increase in dopamine. In red, I'm plotting the dopamine dip caused by stimulation of GTACR, which suppresses the neuronal activity. With this inactivation protocol, we saw that D2 receptor SPM-PK activity showed marked increase. And this strongly suggests that there is a basal binding of dopamine to D2 receptor, which allows the D2 receptor PK activity to increase at the time point of dopamine dip. Furthermore, we confirmed that this optogenic perturbation indeed was dopamine receptor dependent by pre-treating the animals with either D1 receptor or D2 receptor antagonist. In both cases, the receptor antagonist was able to remove the PK activity modulation for D1 receptor SPNs and D2 receptor SPNs. So in summary, I have shown that dopamine receptor is necessary for SPM-PK modulation that is correlated with dopamine release pattern during behavior. Also, the activation and inactivation of dopamine neuron activity is sufficient to recapitulate the pattern of PK activity observed during the behavior. More specifically, D1 receptor SPN's PKA really only responds to dopamine increase, while D2 receptor neurons PKA only responds to dopamine decrease. With this information regarding how dopamine is modulating PK activity in vivo, we can now come up with the following model. When dopamine neuron activity increases because of the reward or optogenic activation, that increases the dopamine level above the baseline, and that dopamine can now bind to D1 receptor, therefore increases the PK activity. On the other hand, for D2 receptor, that's already bound to basal dopamine, this extra increase in dopamine does not really affect the D2 receptor, therefore it does not really change the PK activity in D2 receptor expressing neurons. The situation is flipped for reward omission or optogenic inhibition that decreases dopamine below the baseline. D1 receptor is mostly unoccupied by the basal dopamine, and therefore the dip in dopamine does not really affect the PK activity in D1 receptor expressing SPNs. On the other hand, the D2 receptor is bound to basal dopamine. Therefore, the dip in dopamine now allows this basal dopamine to unbind from D2 receptor, and therefore increase the PK activity in D2 receptor SPNs by this inhibition. Lastly, I wanted to test if the PK activity was actually important for learning. To test this hypothesis, I relied on overexpressing the endogenous peptide inhibitor of PKA called PKI. So let's quickly go over what PKI is. PKA, the actual enzyme, is a multi-subunit enzyme consisting of regulatory subunit depicted in gray and catalytic subunit depicted in green. When cAMP binds to regulatory subunit of PKA, that allows the catalytic subunit from unbind from the regulatory subunit, then catalyze the substrate or phosphorylates the substrates. PKI works by mimicking the function of regulatory subunit. In other words, it binds to catalytic subunit of the PKA and suppresses the activity. And we can now take advantage of this highly specific inhibitor of PKA, which is a peptide, by overexpressing the PKI using AAV in a cell type specific manner. We confirmed that PKI indeed blocks the PK activity in vivo by co-expressing FLIMACAR and the PKI in D1 animals. To have a relatively strong expression of the sensor before the expression of the PKI, we first injected the FLIMACAR virus 2 weeks before the PKI virus injection, and then performed the longitudinal experiments of checking the response of D1 receptor SPM PKA activation in response to D1 receptor agonists across 12 days. And we can see that PK activation of D1 receptor agonists is reduced as the expression of the PKI increases across days. With this tool, we tested if the inhibition of D1 receptor SPM PKA has an effect on learning itself. And in this graph, I'm plotting the test group injected with PKI in red, and the control group injected with GFP virus in black. Here I'm plotting the time in trigger zone, the speed after LED, and entering failure rate that are behavior estimates of learning. And you can see that PKI, the red group, although it does learn eventually, has a slower learning, especially in the early phase of learning, which is consistent with the activation of PKA in D1 receptor SPNs in early phase of learning. The trend was much more subtle for D2 receptor SPN's PKA inhibition, but was reversed. There was a minimal difference in learning in the early phase of learning, but there was a small change in the late phase of learning. This is consistent with the D2 receptor SPM PK activation in the late phase learning during the failure trials. To conclude, using fluorescence lifetime photometry, optogenetics, and cell type specific inhibition of PKA using PKI, I've shown you that how dopamine can act as a teaching signal through the SPM PK activity. Furthermore, this allows us to reach the two large bodies of neuroscience studies, the studies that focus on what dopamine can encode and do, and the studies that focus on what PK can do in terms of neuronal plasticity, and provide us an insight into how plasticity could be modulated by environment in real time. So what are the implications of study? Today, I've shown you that we now have tools to measure both neuromodulators and intracellular kinase activity in behaving animals, which used to be restricted to in vitro experiments. And by bringing these tools to models of disease and comparing their patterns in health and disease, I think we may be able to better understand the pathophysiology as well as identify the possible molecular targets for treatments. Also, in more mechanistic terms, as we have seen in the dopamine-dependent modulation of striatal neurons' PK activity, different baseline levels of neuromodulators can really significantly affect the downstream pathways response, and therefore non-physiological manipulations of neuromodulator may result in unintended consequences due to the sensitivity or insensitivity of the receptors to endogenously released neuromodulators. So with that, I want to acknowledge my thesis advisor, Bernardo, who gave me amazing scientific experience for the past 4 years. And I also want to thank my collaborators and funding source. And I want to thank everyone for paying attention, and I'm happy to take questions.